Hello and welcome to my channel Sam Biology Class. Friends, today we will discuss about bacterial transposons. But before that, will you please consider subscribing my YouTube channel? Your subscription is often a lot to me. Transposons are mobile genetic elements. They are present in most of the living organisms, including humans. Contrary to the previous view that genes remain fixed at their unchanged position on the chromosome, the genetic elements are capable of moving or jumping from one location to another within the genome. Hence, they are also known as jumping genes. The movement of transposon from one location to another is known as transposition. Bacterial transposons are DNA segments which can relocate themselves into different positions within the same bacterial chromosome or are transferred to plasmid and bacteriophage DNA. They often carry extra gene, most notably for antibiotic resistance. For example, beta-lactamase gene. This makes the treatment of bacterial infection difficult. They also contains the gene for the enzyme transposase, which is involved in transposition. Transposon facilitate genomic rearrangement, cause insertional mutagenesis, alter gene expression, and drive evolution in bacteria. Most importantly, they also contribute to the spread of antibiotic resistance. Transposons are also known as jump DNA and selfish DNA. Bacterial transposons are divided into 4 groups. Number 1, insertion sequence element. Number 2, composite transposon. Number 3, non-composite transposon. And last one is conjugative transposon. First one is insertion sequence element, also known as insertion sequence or insertion element. Insertion sequences are the smallest and simplest transposable elements that are predominantly found in prokaryotic genomes. These mobile elements are 700 to 2500 base pairs long. Insertion sequences have 2 major characteristics. Number 1, they are small relative to other transposons. And number 2, they carry only the gene for the protein transposase, which is implicated in transposition. Insertion sequence elements are named according to the form ISN, where n is the number. Examples are insertion sequence 1, insertion sequence 2, insertion sequence 3, insertion sequence 10, and so on. Regarding structure, each insertion sequence consists of a central coding region which is flanked on either side by short inverted repeats. These are the 2 inverted repeats, and this is the coding region which contains the gene for the protein transposase which is involved in transposition. Second one is composite transposon, which is represented by the symbol Tn. These mobile genetic elements are larger and complex than insertion sequences. Composite transposons are from 2.5 kilobase pairs to 60 kilobase pairs long. Each composite transposon consists of a central coding region which is flanked on either side by insertion sequence elements which may or may not be identical. And the coding region contains gene for antibiotic resistance, for example for resistance to kanamycin or tetracycline or chloramphenicol. This is the structure of composite transposon TN10 in which the coding region contains the gene for tetracycline resistance, and the central region is flanked on either side by insertion sequence 10. Hence, the transposon is named as TN10. The inverted repeats which are present in these insertion sequences are recognized by the enzyme transposase. As a result, the complete genetic element is transposed as a single unit. Composite transposon transpose from plasmid to bacteria and also between bacteria, thereby contributing to the spread of antibiotic resistance. Third one is non-composite transposon, which is also represented by the symbol TN. Non-composite transposons are from 1.3 to 6 kilobase pairs long. These mobile elements consist of a central coding region which is flanked on either side by short terminal inverted repeats. These are the two short terminal inverted repeats. So in these mobile elements, insertion sequences are absent, and the coding region contains genes for enzymes such as transposases and resolvases, which are implicated in transposition. In addition, non-composite transposon often carry gene for antibiotic resistance. For example, in TN3, the antibiotic resistance gene code for ampicillin resistance. Non-composite transposons also contribute to the spread of antibiotic resistance and other traits, thereby aiding in bacterial evolution. Last one is conjugative transposon, which is also known as integrative and conjugative elements. These mobile DNA sequences are integrated into the bacterial chromosome that promote their own excision. The excised transposon is then converted into a circular intermediate, which is then transferred by conjugation. These mobile elements facilitate horizontal gene transfer and also contribute to the spread of antibiotic resistance across broad diverse bacterial species. Examples are TN916 and TN1545 family, which is prevalent in gram-positive bacteria. These mobile DNA sequences mediate their own transfer by using a mechanism similar to a plasmid-promoted conjugation. Finally, bacteriophage mu, which is a temperate virus, also act as a gene transposon that integrate into the host genome in order to initiate its life cycle. Friends, this is all about different types of bacterial transposons. For more updates, please subscribe my YouTube channel, Sam Biology Class.